Okay, so this is the review sheet for DNA and inheritance. So let's have a quick look through our answers. So first of all, we have then in our cell, from the cell then, we're going smaller. In the cell then we have a nucleus. Inside the nucleus we have chromosomes. On chromosomes we have 46 pairs, as you remember. Chromosomes then are broken down into DNA. It's made up of DNA. Then if we take a section of DNA, we will get a gene that will code for a characteristic of ours. So DNA consists of two long chains. The long chain has alternating sugar and phosphate backbone connected by our bases, okay, and is twisted into that double helix, okay. It is that double helix shape. So if we're going to draw DNA, it is that double helix shape. The order of the bases makes proteins from amino acids, okay? And that then is called the triplet code, okay? And it's there because 3 bases will code for an amino acid, which will make a protein. We then have the 4 bases. We have A, T, C, and G as our 4 bases. Adenine, I'm going to write it this way. Oh, no, I'm not, because I haven't got space, so I'm going to work it out. Nah. So we have adenine, thymine, guanine, and cytosine, okay, as our 4 bases. You will need to remember the name of those 4 bases to get a mark. If in the exam they ask, you will need to know the name. You can't just give A for adenine. So then we have the complementary base pairing, where A bonds with T always, okay, and then G bonds with C always. So then for you, then you had to find the alternate code. So if we're looking then, if we go closer, we have A, so we would put T for our alternating code. And we've got T, so A, G to C, C to G, C to G, A to T, T to A, G to C, A to T, T to A, G and C, okay, just given our alternating. Then we've got, describe the process of genetic profiling, okay. So first of all, what you would do, you would isolate, okay, you would separate the DNA from other tissues, okay. Then we would use a process of fragmentation. So we'd use an enzyme to break down the DNA into short lengths. Into shorter lengths, remember DNA is a really, really, really long molecule. So we cut it down into shorter lengths, okay. Then we separate it, okay, through gel electrophoresis, okay. So essentially then we have the DNA down one side and we pass an electrical current across a layer of gel. The fragments of DNA will move across different distances depending on the weight of the DNA strand. The lighter the strand, the further across the gel it will move, and the heavier the strand, the shorter it will move. Okay, then what you would do then, you make a comparison and you would compare the DNA bands with other samples. Okay, so why do we use genetic profiling? Okay, we use it to determine paternity, normally in paternity tests, and to solve crimes. If there was DNA left at a crime scene, we could compare it to a suspect to see if it matches their DNA. Okay, then we have what is genetic modification. That involves taking genes from one organism and placing them into another. So we take a gene out that we want. So we've tried this on insulin. So we take the gene out that produces insulin and we put it into then like say a diabetic, and we replace their faulty gene with the new correct gene to hopefully try and cure their diabetes. What are the advantages then? Well, we get disease-resistant plants, mainly, so GM crops, and we produce a higher yield of crops if they have disease-resistant genes in them. Disadvantage then, we limit biodiversity. And if you're not sure what biodiversity is, it's the number of different species in a particular in a particular environment. Okay, and let's go up. Keywords we need to know. So what is a gene? Gene is a small section of DNA that codes for a particular, I missed the word, characteristic. Okay, then we have alleles. So an allele is a different version of the same gene. So for example, brown eyes, blue eyes. A dominant gene then controls the characteristics of the organism, and we represent that by a capital letter. A recessive term then, it only controls if the individual has 2 copies of it. So in that case then they would have 2 lowercase letters. So for example, little b, little b. Then we have heterozygous, so our alleles that are different. Then we have homozygous, so our alleles that are the same. We have a genotype then. Genotype then are the genes an organism possesses. Okay, it's a set of alleles that they have. So is it big B, little b, big B, or little b, little b. And the phenotype then is the physical characteristic of the organism. So what does it actually look like? Does it have brown hair? That would be a phenotype. Blue eyes, etc. Then if we're going to draw a Punnett square, we have draw a Punnett square for a homozygous dominant male and a heterozygous female. Of blue eyes, we've got B is dominant over brown eyes. Okay, so if we look here, then the homozygous dominant, okay, because it's homozygous, that our alleles are the same. So it's either going to be big B, or little b, little b. Because it's homozygous dominant, okay, it makes our male big B, because we've got dominant, so we need a capital letter, and we're homozygous, so they must be the same. So he is big B. Then our female then is heterozygous. Heterozygous means our alleles are different, so she must be big B, little b. And we set out our Punnett square, literally noughts and crosses box, and then we separate our alleles. The matter which way the male and the female go, just that we split them, one goes on each side. One goes top, one goes on the side. Okay, and then we pair them up. So we have big B, big B, little b, big B, and then little b, big B. Okay, and then work out the eye colour of the offspring. Well, 100% would have blue eyes. And the reason is that every single offspring has a capital B, okay? It has a dominant allele. And we know from the question that blue eyes is dominant over brown eyes. So all the offspring would have blue eyes. Okay. Then we've got Explicit Cross, which determines the sex of a species. And we know then that the male chromosomes are XY and females are XX. You would set it up the exact same way we just did. So I've got female on top, XX, then I've got male across the side, XY. And same thing again, we take our 2 and we put it into our box. We've got XX, XX, XY and XY. So we are literally then 50-50 on having a boy or a girl. I think that is it for our review sheet.